Hello, wonderful people. Here we are, back studying Mendelian genetics, this time chapter 13, where we combine chromosomal theory with that of Mendel's ideas of meiosis. So let's put Mendel and modern biology together. This should look familiar. Here we have a cell. Now this is a simple showing of just one type of chromosome. So in this cell, n equals 1, but it's a diploid cell, so it's a 2n cell. And at the same time, it has gone through replication. So it's gone through the S phase because each of these chromosomes has 2 copies of the chromatids. So there are 4 chromatids in this cell, and the chromosomes are all lined up together, homologous chromosomes pair. Now, these are homologous chromosomes. They contain the same genes, but they don't necessarily contain the same alleles, and in this case they do not. Here we have the gene for coloration in a pea. Blue, or daddy, has the little y recessive gene, and red, for mommy, has the big Y dominant allele. So this is a heterozygote, and this heterozygote is going to start to make its own gametes. Well, in metaphase, the spindle fibers attach to the kinetic cores, and they pull apart the chromosomes. Now this is where segregation takes place. Little y is in one cell, and big Y is in another. But we have not produced haploid cells, which is necessary for meiosis, and so it has to go through the second phase of meiosis, the second round of division, and then it finally makes the haploid cells. And we can see that there's big Y, little y, big Y, little y, big Y, big Y. So we have those 4 gametes. But the 2 blues and the 2 reds each contain the same alleles. So little y, big Y, big Y. We have a 50% chance of getting little y and a 50% chance of getting big Y. So here we've painted the alleles on the chromosomes, and we can see exactly Mendel's first law of segregation and how it works with meiosis. Of course, it gets more complicated when we add in more genes. So here we have a dihybrid. Now this should look a little familiar too, because we're talking about the 2 genes that we've been talking about for so long with peas. Color, big Y, little y, and shape, big R, little r. All right, so these are on separate chromosomes, so now we can show that big R does not control big Y or little y, and vice versa. Let's go. Let's figure out which are the homologous chromosomes, which are the sister chromatids, who's paternal, maternal, and what's dominant recessive. Okay, I want you to pause and figure those things out for yourself. 3, 2, 1. All right, back out with you. Hope you figured it all out. Homologous chromosomes are those chromosomes of the same type. So this is a big chromosome and this is a big chromosome, which contain the same genes, including this shape gene for the P. These are the same type of chromosome, smaller chromosomes, and they contain the same genes, specifically in this case, the color of the P. Now they have different alleles, but these are homologous chromosomes. These are homologous chromosomes. This and this are non-homologous. This and this are non-homologous. They contain different genes. Each of these chromosomes represents a pair of sister chromatids. So these two are sisters. These two are sisters, tied together by the centromere. Sister, 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 sister. Here we represent paternal by the color blue and maternal by the color red. So you get a copy of the big chromosome and a copy of the little chromosome for your dad, and you get a copy of the little chromosome and the big chromosome from your mom. Dominant and recessive? Well, the capital letters in this case represent the dominant. So big R for round and big Y for yellow. I know you got that right. All right, here is an example of the dihybrid cross combining Mendelian genetics and meiosis together to see how it works. We start out with our true breeding seeds. Here we have yellow round seeds and green wrinkled seeds. Dad in this case, mom, excuse me, in this case is big Y, big R, and she has 2 copies of the big Y and 2 copies of the big R. That makes her true breeding from generation to generation. Here we have true breeding recessive from dad, little y, little y, little r, little r. When these two are crossed, they produce gametes. Big R, big Y is the only choice here because they only got big R and they only got big Y. Little y, little r is the only combination we can get here. That produces an F1 plant that is dihybrid or heterozygous for both genes. Now we cross 2 F1s together to try to produce the F2s. And here we see at this stage we have the big R and little r and big Y and little y. They have got to match up because these are the homologous chromosomes. Homologous chromosomes. Homologous chromosomes. Homologous chromosomes. And they form what we call a tetrad. It is also referred to as a bivalent. Now, please remember that these chromosome pairs now line up head to toe, toe to foot. Head to toe, toe to foot. In anaphase 1, the alleles now separate equally so that half of the alleles get big R, half get little r, half get big Y, half get little y. That, my friends, is the first law of segregation. Now we can see the law of independent assortment as well. How is it that big R and big Y do not control each other? Well, it all comes down to how these chromosomes are arranged on the metaphase plate. So please take a careful look at this. We can see that big R and big Y are lined up on the same side of the metaphase plate. They are going to travel together, just as little r and little y are going to travel together to the new cell in anaphase 1. But that's not the only way that the chromosomes can line up on the metaphase plate. They can also line up so that little r is with big Y and big R is with little y. All we have to do is switch these two around and we get this arrangement. Now we'll have little r and big Y and big R and little y traveling together. At metaphase 1 then, we set up the law of independent assortment. All right, so this is the same picture, only it's showing us the second end phase as well. So what happens as we go through the law of segregation in anaphase 1 and law of independent assortment in metaphase 1? Well, at the end of the day, we have our 4 different types of gametes that are produced. Big Y, big R, little r, big Y, little r, and little y, big R. We can see that as a result of meiosis, we would end up with 2 of each out of 8. That gives us a ratio of 1 to 1 to 1 to 1, or 1/4 of each. And that is exactly what Mendel predicted. This is showing the same thing in just a slightly different way. All right, folks, what I'd like for you to do is to go ahead and pause this video and then write down again all of the gametes that are possible and all of the phenotypes, and make sure that you get that 9 to 3 to 3 to 1 ratio, or as written here, 1 to 9 to 3 to 3. Okay.